Time Center crews boldly go not to distant galaxies, and they don't rely on a warp drive. They use cutting-edge microscopy and optical measuring techniques to explore another universe. Operating at nanoscopic level, they look at individual cells not visible to the naked eye, and they identify, monitor, and capture processes within cells.
High-resolution fluorescence microscopy is a powerful technique for observing specific molecules like proteins or drugs in living cells or tissues. It allows us, for example, to study dynamic processes like the invasion of a virus into the cell. This enables us to understand better how diseases progress and to develop effective treatment therapies.
Light microscopy allows the characterization of blood cells in real time. By looking at the cell morphology, protein movements, and cluster formation, researchers can determine the state of blood and thus the quality of transfusion blood supplies. Super-resolution microscopy is a modern technique used at Time Center. It is state-of-the-art in only a few medical studies around the world, but it will fast become the gold standard in light microscopy.
The super-resolution microscopy allows us to image cells and tissues in 3 dimensions below the resolution limit. This extremely high resolution, which is less than 1,000th of a human hair, allows us to make discoveries in areas of virology, immunology, and neuroscience. Super-resolution becomes one of the fastest growing techniques in our laboratory.
The researchers can study in 3D, for example, how a thrombus, that is a blood clot, is formed, or how cells interact with 3-dimensional cell scaffolds that mimic an artery. Looking deep into the building blocks of life helps Time Center to answer fundamental problems of modern biology and medicine.